As part of Chapter 2, we're going to do a brief discussion on microbes in the lab. And basically, we're just trying to get an understanding of how you culture microbes in the lab. So when you're culturing microbes, especially bacteria in the laboratory setting, there are 5 steps that are generally used, and we're going to refer to those as the 5 I's. Why? Because each starts with the letter I. So you have inoculation, incubation, isolation, inspection, and identification. So let's start with the first step. Okay, when you first start cultivating microbes, you have to obtain a small sample known as an inoculum. And then what you're going to do is introduce that inoculum into the new environment, which we're going to grow that microbe. In the lab, we usually use a medium such as an agar plate or a broth test tube to grow that microbe. Now, the process of taking that inoculum and introducing it into that new environment is known as inoculation. Now, just so you understand, in the clinical settings, you might get your inoculum from the feces, saliva, blood, or any other human material, okay? After you've taken that medium and you've inoculated it, you've got to give it time to grow. So when you're growing your microbes, you have to take into consideration what is it that is their optimal environment, because you want it to grow and you want it to grow well. So when you're talking about incubating, you have to think about things like what's the temperature, what's the O2 and the CO2. Most things that we're going to be dealing with are going to be we're going to be looking at growing them at 37 degrees Celsius, because those are going to be microbes that can live and grow well in the human body. After incubating your microbes, the growth you see on the plate or in the broth is known as a culture. And cultures are classified by the number and the type of species present. So if the culture has only one type of microbe from a known origin, it's known as a pure culture. If there are 2 or more microbes that are identifiable, then we refer to it as a mixed culture. However, if you have a culture that contains an unknown or even an unwanted microbe, then we say it's contaminated. But when you start looking at a culture, there's going to be many times you're going to need to separate out a mass or a group of cells, and we call those a colony, and you want to separate it out from others. This is especially true when you're using like human specimens for your inoculum, because when you do throat swabs and things like that, they're going to have all kinds of microbes, and you want to isolate out one. So to separate one colony from a whole group is known as isolation, and there's going to be 3 different methods to use. We can use a streak plate, pour plate, or a spread plate. So if you look here at this diagram, the first row is with the 3 plates. That is the streak plate. Now, streak plates can be done in either 3 or 4 sectors. You grab a bacterium from your growth plate and you streak it in one area. You turn your plate and then you streak it again and do it 2-3 times to give you a total of 3 or 4 sectors. Each time you're doing this, you're diluting the sample. Underneath that, down here at the bottom, you have the pour plate method, and that's a serial dilution method is used by diluting the specimen with each subsequent tubes of broth. So you keep diluting and diluting, and then the tubes are poured into a petri dish and allowed to cool, and then the agar will solidify, and the colonies are going to go through the medium and they're going to grow up to the top. The third one is known as a spread plate method, and in that you take a known amount of broth contaminated with the specimen and then you just pour it on top of an already solidified agar plate, and you use a spreader and you spread it all the way across evenly across the top of that, and that dilutes it across the surface. So these are methods for isolation. So after you've incubated your inoculum and you have after that isolation and you've incubated it again and allowed it to grow, now you're going to do this inspection process. And when we inspect, we have to do it in 2 ways. First, we must inspect our culture macroscopically. In other words, you're just going to use your eyes. You can learn such a great deal of information from macroscopic information. In particular, when you're looking at your plate, you're going to make sure that you have growth and make sure that you have if you're looking for it to be a pure culture, that you only have one type or one colony growing. So do they all look of the same color, shape, size, things like that? If not, if you end up having more than one look to your colonies, you might have to go back and repeat the last step and do another isolation process. But you're going to first look macroscopically to make sure that you've got to what on the plate with the naked eye looks to be one colony or a pure culture. Once you have that, then you're going to go to the microscopic inspection, and here you're going to have to use staining processes. So you're going to take a fixed sample, you're going to put it on a glass slide, you're going to stain it for better viewing, you're going to use that microscope under high magnification, and you're going to inspect it microscopically. And here what you're going to be doing is looking to see, do all the cells look to be similar? Are they the same shape, size, other morphological characteristics which help you to determine the purity of the culture? Again, if you've got a mixed culture kind of showing here on the slide, you may have to go back to that isolation process, do another streak plate, do another pour plate, etc. Go back in, macroscopically inspect it, and get here until at this level it looks to be a pure sample. So then once you get through that process, then the purpose, of course, of the 5 eyes is to identify the microbes. So after you have grown it and you believe you got a pure sample, then you got to go through that identification process, and that's generally going to be done through biochemical and physiological methods. Okay, so in many cases you're going to use such experiments or assays known as like the ELISA, DNA analysis, etc. And through those processes you're going to be able to identify that specimen. So just in review, the 5 steps for culturing microbe are: step 1 is for inoculation, the process of introducing a microbe to a new environment. Step 2 is going to be incubation, giving that microbe the proper environment to grow. Step 3, you're going to isolate. This is the process of separating microbes from a mixed population. Step 4, you're going to inspect, and that's going to be both that macroscopic and that microscopic inspection. And finally, step 5, you're going to gather data through both the biochemical and physical assays that you're going to perform, and through a series of those you're going to be able to identify your unknown microbe.